Okay, so thanks so much for inviting me to give this talk. It's like such a good large group. I'm impressed for, a Thursday night, 7 o'clock. So I really appreciate you all for coming. And I'm I'm definitely going to have opportunities for questions throughout. If something pops up and you really want to ask, don't worry about it. Totally do that. And I've also got some areas where I want you to put some stuff into the chat, and I can see chats. They just pop up and they pop out. So I can't see them for super long. So Bronwyn will help to curate or help me see those. So I just put this slide up at the very beginning so you can see, some qualifications for why I'm here before you today. I just want to show some pictures of some of my past. I did my bachelor's degree at Macalester College, also known as the alma mater of Kofi Annan, who's a former UN Secretary General. So he's one of the more famous ones. There's some other authors and stuff, but that's where I did my initial degree. I did my PhD work just down the road at College Park. So I first came to Maryland at that point. I did a postdoc, which is sort of a 2 or 3 year position after you do a PhD at San Diego State University, where I continued to work on topics related to the research that I started as a graduate student. And I've developed sort of my own projects and my own things going on, which has led me to the present where I'm a professor here at UMBC. So this is the sort of landing page of my website. And as you can see, the majority of the research I do focuses on the evolutionary biology of sex. So one of the topics that my lab focuses on quite heavily is why reproduction, as we tend to think of it, which is sexual intercourse, why that evolved and why it's maintained and why it's so common in animals, particularly when there are other ways to reproduce. So that's a major theme of the research I do. And most of that work is focused on arthropods, especially these right here that I'm circling with my mouse, which are from a group called Opiliones. So if you're from the East Coast, which I saw we've got somebody visiting from Florida, and many folks from different parts of Maryland, you've likely seen these, possibly many of these at one time, and you may have looked at them and said, okay, well, I think I know what that is. But in the same way that I think of sort of these images from the famous painter Rene Magritte, who painted this, which is a pretty common image of his work. He was a surrealist painter, so he would paint things like pipe and then say, in French down here, this is not a pipe, right? So it's a picture of a pipe is what he was kind of getting at. When you look at these, I hope in the future you see an Opilionid and you realize this is not a spider. It's actually from a completely different group. So throughout the talk here, I'm going to use some art and myths and sort of legends about these organisms that we on the East Coast at least tend to call daddy-longlegs, and I'm calling them dandy-longlegs because they are male and female, of course, and I'm going to try to dispel some of the typical myths about this group with some facts. So first, I kind of want to go back to the very basics here, like Bronwyn was saying, and ask what people's favorite arthropods are. And if you don't know what an arthropod is, yes. So let's have a second or two where folks put the name of their favorite arthropod into the chat.
That's so hard. There's so many.
Yeah, right. It may be easy or hard. I don't know. I suppose I guessed a lot of people would probably say, periodical cicadas, because boy, did we have an amazing summer. Awesome. I'm seeing some people say blue crabs, millipedes, a lobster, delicious and cool. Ladybugs, jumping spider, Lucas the jumping spider, a very specific jumping spider. dragonfly, praying mantis. We've got so many orders of arthropods represented here. Garden spiders, got some, I'm doing some research now. Fireflies, sadly, sort of like not as common as they used to be, but awesome. So I'm so glad to see so many different suggestions of favorite arthropods, which indicates to me that everyone's already pretty familiar with what an arthropod is. So arthropods are a phylum, so it's a group of organisms, a pretty sort of broad group of organisms that includes everything that has jointed legs and a sort of a hard exoskeleton. So that's going to include, of course, the crustaceans, the insects, the millipedes, and the centipedes, and this group called the Chelicerata. So this is a phylogeny. This may be sort of an unfamiliar way of looking at different groups of species, but this is what we typically use when we're thinking about the evolution of species, is sort of a family tree. So who's most closely related to who? So this group of arthropods is part of a broader group called the ecdysozoans, which basically refers to the fact that these things all shed their skin. They go through ecdysis or molting, and they include some things that don't have jointed limbs, like nematodes, sort of flatworms in the soil, tardigrades, you may have heard of those. We also call them water bears, onychophora or the velvet worms, and then of course the arthropoda, which we've also already talked about. So not only do the arthropoda shed their skins, but they also have jointed legs. So now we've talked about sort of the broader scope of where these Opiliones or daddy-longlegs sort of fall in the group of arthropods and ecdysozoans. So what are daddy-longlegs specifically? This has always been an interesting question to ask around the country because what is considered daddy-longlegs is very different depending on where you are. So the important thing I want to show, oops, sorry, I skipped ahead a little fast, is that daddy-longlegs are part of a group we call Opiliones or harvestmen, which I'll talk about where those names come from, that is completely separate from spiders. So not spiders, spiders. And so they're separate from spiders in the same way that scorpions are separate, solifuges are separate, the Telephonida or the whip scorpions and amblypygids are separate. So all of these are separate orders of the Chelicerata are within arachnida. So all of these are arachnids, but there's a lot more groups than just the spiders. And I put some of the characteristics that we see in a lot of these things. When we look at the phylogeny of just lineages within their arachnida, so that's just everything within this dotted box that extends all the way down, you can see the araneids are a pretty big group. At the root of this phylogeny are species that diverged a long time ago or have very different genetic sequences. And so Opiliones is sort of close to the root of all of the arachnids, meaning that this is a group that diverged a pretty long time ago. It's quite ancient, and it's actually pretty speciose. So here, these are just examples from these different groups. Opiliones in this paper was suggested to be most closely related to what are called hooded tick spiders. So they're kind of an odd little group called Riciniulae. But basically they've been around a really long time, and there's actually quite a few species of them. And the only groups that have more species are the mites and ticks and the spiders. Okay, so now we're gonna play our next game, which is where I ask you which ones of these pictures are Opiliones. So I want you to look at each of these 6 pictures, and I want you to put into the chat which numbers, 1, 2, 3, 4, 5, or 6, or all of them or none of them are Opiliones, you think. And yep, I see now I'm looking at the chat. I see there was a good question a little bit ago about ticks, whether they were spiders or also another group. They are a separate order of arachnids, just like Opiliones and scorpions are separate orders. So they're not spiders. And I'll show some of the features that they do have in common within arachnida. So you'll be able to see that a little bit better. So a lot of answers are coming in. A lot of folks think number 4 down here, an Opilionid. 2 is on some people's minds. 1, 3, and 6 have been guessed. 4 again, 6, 4. So a lot of people are very convinced about this one being an Opilionid. Very interesting. Not seeing too many 3s. 5 has been guessed, so 5's not a yes. Okay, so I think we can probably now, a lot of people have put in their guesses, and so now I'm going to go through which ones of these are Opiliones and which ones are not. All right, so numbers 1 and 3 are not part of the order Opiliones. Number 1 is a true spider. It's part of the Araneids. It's a stellar spider. The genus is Fulcus. So these are the ones that you're likely to see in your shed or in your garage. They build cobwebs and they like to shake to sort of frighten off potential predators. Sometimes they're okay being near webs of other conspecifics, other individuals of the same species. So they're okay being sort of social. And the reason they tend to be lumped in as daddy-longlegs is because they've got these long, skinny legs, but they are actually a different order. They're true spiders. Same thing goes for this one, number 3. They're often called mosquito hawks or crane flies. So this is an actual fly. You can tell because it has these little halteres here, those little balancing organs. So it's got just 2 wings. To my knowledge, no arachnids with wings have ever been found. So that's a pretty good indicator that this is not an Opilionid, although they are sometimes called daddy-longlegs because, again, they've got the long, skinny legs. All right, so these are both very common. You know, if you go out in a park or, you know, you're moving things around in your shed, and you're likely to come across one or both. And so the others, 2, 4, 5, and 6 are all Opiliones. And so I gave you some kind of hard ones, right? Because number 4, this is from a genus Liabunum, which are very common in temperate areas. So these are super common here. And so you've definitely seen them, and a lot of people were really confident about these being Opiliones. But actually numbers 2, 5, and 6 are also from other suborders, or they're from other groups of Opiliones. So this one comes from New Zealand. It's got these really long chelicerae. So these are actually its mouth parts that are so long. This is a very small one. I should have used a scale bar here, but as you can see, it's got really big eyes. So this species, Cato agilis, does occur in Eastern North America. It's quite small though, so it might be easy to overlook, but it tends to like to hang out in mossy places, and it uses its pedipalps. So these are not the mouth parts. These are actually kind of like arms and hands that it uses for feeding. And it chases after small prey in the leaf litter and on the ground. This species here is Cyroexilus. It's also from North America. And this one may look like a mite or a tick to you. And it lives in sort of a similar way, but it doesn't have sort of piercing mouth parts or stylets. It like all of these, has little scissor claws that it uses for cutting up its food. So these would be found in the leaf litter as well. A whole host of body forms that are shown in the Opiliones. And so let's see if I can get this to go away here. This bar at the top, because I know it might sort of interrupt your ability to view things. There we go. Okay, so these are just names for Opiliones found found in countries all over the world. So as it turns out, Opiliones is like a very, very wide ranging group. We find them in temperate and tropical environments. And the only place we don't find them is in Antarctica. So, and they actually can tolerate pretty cold temperatures. So they're found in, some mountainous or cave-like habitats, as well as in tropical rainforests, and of course our temperate forests in North America. So all across the globe, we see a lot of different names, common names for these organisms. Most of them have to do with the fact that they've got real long legs, or that they tend to be really common during the harvest time, which is typically in the fall. In a number of countries, such as Ukraine, Russia, the name for Opiliones has to do with with the fact that it tends to be around during harvest and is called reaper, or hay reaper. So that's what kosar and senokosek means reaper, like for wheat. In both China and Japan, the name for Opiliones has to do with the fact that when they walk, they tend to move their first legs or second pair of legs around, almost like to tap, like a blind person might use a cane. So zatomushi in Japan actually means blind bug. And so of course we know it's not a bug, but zato is the sort of operative here. It's referring to the fact that it looks like it's walking around like it's blind. Mangshu means the same, it means blind spider. Probably the country that has the most common names for daddy-longlegs or Opiliones would be Brazil, which has a great deal of common names. Frade Federento actually means stinky friar, smelly friar. And Chinchina, which comes from Argentina, means smelly bug. Both of these refer to the fact that, as I'll show you in a minute here, most Opiliones have sort of these glands on their back that produce a smelly fluid, which is repugnant to predators. So it's one of their defenses. So these are carvings from first century Rome of different herders with some livestock. So this is actually where the Opilio and Opiliones comes from. So an Opilio was a shepherd during Roman times. And so the Opiliones is Latin and actually means shepherds. And the reason that probably got used is that Latin is typical for naming species and groups. But we know that there were this various periods, different shepherds that would use gear like these sort of hooks and stilts in order to keep track of their flocks. So this was in the Landais, which is like Western France. So they would get on these stilts in order to see over the rolling hills where their flock was. And they actually could do all sorts of things. They could run and do feats of true acrobatism, as was said by Scientific American in 1891. So this is probably why that name got sort of chosen, Opilio, having to do with shepherd. So these long legs. And also the harvestmen or harvester epithet comes from the fact that they tend to be around or more noticeable during harvest time when people are out in the fields. So there's various legends associated with the fact that they tend to be seen during harvest time. In fact, in England in the 1600s, it was believed that seeing Opiliones was a good lucky sign, and you should never ever kill one because they actually would help you harvest your crops at night. And so it was believed that they were lucky to have around because they were sort of like little fairies that would thresh wheat at night when no one was around to see them. So in France, similar thing. During the evenings, seeing an Opiliones was good luck. And the Pennsylvania Dutch also thought it was useful to have daddy-longlegs around because if you picked up their body with all their legs, but one, the free leg that was sticking down, would point towards where lost cattle were. So these are just some various sort of positive legends around Opiliones. One of the main things I want everyone to get from this talk is that Opiliones aren't spiders. So, check, we've already got that one. The other question that I tend to get really frequently is whether daddy-longlegs, Opiliones, harvestmen, whatever you want to call them, are the most dangerous spiders in the world. So you already know spiders is the wrong word to use because they're not spiders. So importantly, I want everyone to know that daddy-longlegs are not venomous. They don't produce silk and they don't produce venom. So they're not dangerous to people. So here is just a breakdown of some of the things that are really similar about the bodies of spiders and Opiliones. They're both arachnids. They both have the same mouth parts, which are called chelicerae. That's where chelicerata comes from. So anything with chelicerae will be part of that group. And they do both have 8 legs. But they do have a lot of differences, which aren't always easy to see offhand. So their mouth parts are very different. So this is what the mouth parts of Opiliones look like. So if you look up close, you'll see they have 2 tiny little crab claws. And so it's almost like using scissors instead of teeth to cut up your food. So they have to cut everything really small, and then they actually swallow that food whole. So they don't liquefy their food, which is what spiders do. They have fluid meals because they use their venom to break down what they're eating. Spiders usually have a waist, so we can usually see some separation between the head and the abdomen. Opiliones don't usually have a waist. They just have sort of one big body part or one little body part. And again, spiders make silk and they make venom. And those are things that make spiders incredibly fascinating. But Opiliones are still really cool because, they've managed to diversify into all these niches. They have all these different unique body plans, but they don't make silk and they don't make venom. So if you're ever out and you see somebody point out some Opiliones and say, oh no, those are dangerous, good thing their fangs are too small to bite us, then you can correct them on all accords because they have chelicerae, so their mouth parts are little scissors. They don't make silk, they don't make venom. Here are those ozophores. So those are the areas where they produce the smelly fluid that some species make. I think it smells kind of like onions. So it doesn't really bother me, but it might bother, a bird or something. So they do exude that when they're being handled roughly. This is just to show their palps. These are a couple of different species that I work with from Japan. So their pedipalps are kind of like their arms, and they use them to handle each other, to grab each other, or to hold their food. And the legs have been removed for these because we use them to extract DNA. So getting back to this whole sort of, are they dangerous, are they scary thing, we've got some art that suggests that they're actually a very good and hopeful sign. So this is a famous Salvador Dali painting. I like surrealist artists. And this one is quite famous because there's sort of a weeping angel and sort of terrifying, things going on in the background. And yet in the midst of things is a daddy-longlegs. There have been books that use daddy-longlegs as sort of an allegorical figure for people who look a little bit different and are a little bit strange and odd. This one talks about a person who moves into a town and he's very strange because of his long skinny legs. And actually this sort of body type was also described in this pretty famous book from 1912 that's called Daddy-Longlegs. So this is a Korean cover that I thought was kind of neat looking. This book is about a girl who grows up in an orphanage, and so she has a patron who pays for her to go to school and pays for her clothes and things like that. And then it turns out it's this sort of odd looking man that she had met in the past, who's been, sort of quietly supporting her that whole time. Okay, so with this picture here, I'm just showing an example of what daddy-longlegs look like when they molt, which, as I said before, because They're part of the ecdysozoans. They have to molt to grow bigger. So these are actually old legs back here. That's the old skin that this individual is growing out of. So these legs look so white because this individual is just molted. And so these are actually pretty soft, and the cuticle hasn't hardened quite all the way. So it's kind of a neat picture that one of my graduate students took of an individual going through its molt to be an adult. So if anyone has any questions thus far, I'd be happy to entertain those now.
Laura wants to know how many species are in Maryland?
How many species are in Maryland? So of the daddy-longlegs type, so of the type sort of like this that you're more likely to see without kind of trying hard to go through leaf litter and look for them, 35, I believe. And if we add in sort of the leaf litter type ones, that would be an additional 5 or so. So 40 species, and they're not all very easy to tell apart. There was a paper published by my former PI, so there's not that many people working on Opiliones worldwide, and I pretty much know them all. But for Maryland, we do have a good key that Jeff Schultz, my PI or my PhD advisor, published. And so to use that key, you do need to know some stuff about the body parts, the morphology of these different species. Sorry, and so that slide that I showed previously is like the kind of stuff you would need to know to be able to tell different species apart. But some of the really common ones are Libionum vittatum, which has striped legs, very, very common in and around Baltimore. Libionum brachiolam is a small species that's bright orange. It actually wasn't described until the 90s, which is interesting because it's so common in Maryland. But it tends to like sort of bushes, edge habitats like near parking lots and stuff. And they can be real numerous and in the mid to late summer. So those are a couple of the species that you're likely to see in just your everyday life.
So there could be more out there, though, if we're all looking. And again, science is not static; it's always moving. So and a lot of discoveries are made by just amateur people out there looking. So keep looking, everybody. Laura wants to know, when they molt, can they regrow legs?
No, so that's a great question. And that's something that is kind of unique to Opiliones. So you may not be able to see it super well. Well, no, these are pretty good. So do you see these sort of break lines here? So this is like the joint. This is the trochanter. Just like on our hip, we have a trochanter, which is what attaches our leg to our hip, basically. So we use the same terminology for arachnids. And so there is a break line here. So if, for example, this individual is out in its habitat and a predator, a snake, grabs the leg and tries to eat this individual, it can actually release this leg, and that's called autotomy or self-cutting. So as far as I know, all of the sort of daddy-longlegs type fauna are capable of autotomy. And it's what they use as a defense, right? So it's kind of a last resort safety thing. And you may notice if you try to grab an Opilionid, I have a sort of method for handling and grabbing them that works pretty well. But if you grab one by the leg, it may pop that leg off. And it's not you pulling super hard. It's them actually releasing their joint in order to get away from you. So they cannot regrow legs, but they can function really well. In fact, their walking is not really affected until they lose 3 or more legs. And we do regularly find them out in the environment, having lost legs and still hanging out.
And Jenny asks, how long do they live?
So that's a great question because it really depends on what species and where they are. So by and large, tropical species tend to live longer than temperate species. So all of our native fauna to Maryland, all of the Opiliones found here will overwinter as an egg or as a subadult. So their adult stage is limited. So they typically live 1 year. In the tropics, they may live 2 or 3. And we think that's probably due to the fact that, as great as they are at handling sort of moist and cool environments, they can't really handle winter, and they've adapted to a life where, they do all of their reproduction in the summer and then they die off. In the tropics, there's not as much seasonality and there's a lot more water. It's more moist, and there's more resources, more things for them to eat. So they can live quite a bit longer. And that seems to be the case even in captivity. Like, we collect them in order to have them lay eggs, and they, the adults just naturally senesce in the fall. So
we have a couple more. We have a question. You may be going to be covering it, and you just talked about it. Do they lay eggs? How many, and how do they mate? And then also, can they smell?
Yeah, I can answer those really fast. So I'll show you what mating looks like, because that's been a pretty significant part of my research. They do eggs. Typical clutch size is 40 eggs or so. They tend to be very susceptible to mold. So I'll show you some examples of species in the tropics that actually have male parental care. The fathers take care of their clutches, of their eggs, and we don't see that really in any other arachnid. So that's a really rare behavior that is evolved in this group. And do they smell? They do have olfaction. So all of their sort of sensory, or the majority of their sensory organs are located on their legs. So they're using their legs for a lot, for, feeling pressure, temperature, and to some degree for smell. So we know that some species seem to go through behaviors where they drop pheromones. They rub their legs on plants to identify territory. Some have sort of swellings in their fourth leg that look like it's probably a gland for producing pheromones. It hasn't received a lot of study because most of the species that have that enlarged pheromonal gland, putative pheromonal gland, are tropical. But we are pretty confident that the majority of their life is a very tactile existence. So they really need to touch things to kind of feel where they are and where they're going. They don't see well. They can probably see light and dark pretty well, but other than that, they don't do much with their eyes, which are those on top of their head there. They just have 2. And yes, so do they taste? I don't know, but they do smell and touch using their legs.
And Tom asks, and maybe you're going to cover it, what is their primary food source?
So again, it's depends on what species. The daddy-longlegs fauna of Maryland are omnivores. A lot of what they eat is dead stuff. So they are pretty happy eating, foraging for and eating dead insects. They will eat each other. They can catch prey. And so they kind of are just, whatever comes across their path, they'll try. I've seen them eat smashed raspberries. I've seen them eat bird droppings. There's a picture online that's been floating around for years of a couple of Opiliones eating a skink tail, a dropped skink tail. So they'll really eat whatever. Like in the lab here, we feed them fish flakes, and they'll eat whatever they can pretty much come across. Some of the smaller species are active hunters and they specialize on collembolans or springtails. So they're eating real small leaf litter insects. And some we think are probably specialists on slugs or snails. But by and large, it seems like they're kind of catches and catch.
Do they stridulate?
They may produce sound. Some species have what's called a labrum. So they have, it's not used for feeding, but they have what looks like a nose that has little denticles or bumps on it. And it looks like they might sort of rub that to make some, we don't really know. They do this stotting behavior where they bounce, which seems to be possibly anti-predator or predator confusion in nature as a tactic. Although it's also been proposed that they tend to do that more when it's cold to warm up their joints. That's something that I had some undergraduates looking to study right before COVID hit. So we haven't really picked up some of those kind of behavioral projects. But yeah, do they sort of actively make sound? Not that we have been able to observe or quantify.
And one more from
Okay.
Simon, age 6. He wants to know what is the minimum number of legs they need to reproduce?
Oh, to reproduce, they don't need legs. So I'll show you, or I think I showed you a picture. So they actually do have intromissive reproduction, meaning that males have a penis and it goes inside the vagina of the female in order to reproduce. So females actually have a storage organ for sperm that's passed from the male to her. And we don't know, we know what those storage organs look like. We don't know how long they're able to store sperm. But the penis is it's something quite important in order to identify species or to identify differences between species, because that's a trait that tends to be very species specific. So they don't need, it would probably be really hard for a male to find a female or vice versa if they didn't have legs, but they don't technically need them for reproduction because they have primary reproductive organs. So I'm guessing Simon probably knows that spiders actually reproduce differently. Males actually charge up their pedipalp, so that their arm. They have a reservoir to put sperm into, and they're able to transfer that sperm to the female's genital orifice using their pedipalp. So males don't have an intromissive organ or a penis. So that is a pretty significant difference between Opiliones and spiders. Great.
There we go.
I'm like impressed with you all. You're definitely keeping me thinking and reminding me about research, plants that have dropped off, prior to COVID. So, okay, so I'm going to continue my talk by talking a little bit about collecting daddy longlegs. And first, you always want an excuse to show cool pictures. All of these are pictures from collecting experiences in different countries. I do quite a bit of my research in Japan, although of course not recently because we haven't been able to travel there. But all of these pictures are from Japan, seeing other sorts of people and animals there. Every once in a while, you run into kids or other adults that are interested in, what we're doing and what we're studying. So that's always a nice opportunity to talk to them. I've been able to take my husband on a few trips with me. And so basically I, force him to be my labor in the field. So basically we can find Opiliones all over the place. Here's just another excuse to show some really neat different morphologies. Here's again a liabunine or a daddy longlegs type Opilionid. This is actually Hadrobunus from Eastern North America. Here's one of my favorite pictures. We call these bunny harvestmen because they have these projections in their abdomen that are probably they're kind of a defense against being picked up. Sometimes we see them with spikes like this individual here, which is a mollusk specialist, we think, from Ecuador. Here is an example of one of those mite-like harvestmen from Australia. Again, New Zealand's got some really cool species that have these really long, elongate chelicera that are probably used by males to show off how sort of big they are. Here's an example of a caddo that we collected and saw lots of in Japan. So there's all sorts of species. And so the collecting methods you use are going to sort of depend on what you're looking for. But generally for daddy-longlegs, you can use active collecting methods. You can tip individuals into a net. You can sweep for them in tall grass. This is a good way to collect Liobunum brachyolum, for example, the small orange one. I usually like to collect specimens by hand, and I like to sort of cage them with my hand. So I like to come over the top of them and then gently sort of pick them up. And that way you don't risk any of the legs being pulled off unnecessarily. You can also do dry pitfall traps if you want to collect species that, especially Hadrobunus, which likes to be on the ground, or small juvenile specimens. They're typically on the ground. They start kind of going up into trees when they molt because they like to be vertical or upside down on surfaces when they do their molting. So if they're dry, if you don't put any preservative or anything in them, you can hopefully collect them live and they'll just run in here. Of course, you need to check those frequently because if they fill up with water or if they're continuing to collect things, they'll start eating each other or they'll die. Wet collection pitfalls use preservatives. Sometimes, depending on what you use, they can be dangerous for non-target wildlife. So you always want to, if you're going to do sort of a wet pitfall approach for collecting arthropods in the leaf litter, for example, you want to make sure you check with your with Natural History Museum or whatever to decide on what are the best preservatives for you to be using. So many people don't know this, but Opiliones are often attracted to black lights. So this is a collection method that you might be familiar with if you've ever tried to collect or see different night flying insects, like near a river or something, because they'll come to this black light and they'll show up in these really cool colors. So they have cuticle that reflects UV in the same way that we see with scorpions. And in fact, because scorpions are mostly nocturnal, this is a great way to, find them. And so the same sort of ideas apply, is that often Opiliones will show up on these sheets, and they'll be trying to hunt insects that are also coming, or they'll start mating or whatever. So it's kind of a cool method to see some arthropods that you might not see as much during the day. And there are many things you can observe about these species, things that we're really just scratching the surface on trying to write about and understand in the different species, such as what they look like as juveniles versus what they look like as adults. So they go through, on average, we think 3 to 4 molts between their sort of baby size that they come out of the egg as and their adult stage. So those are some significant changes in their size, and those molts can actually be really dangerous for them. So those are fascinating to see when we can, but we're just starting to learn sort of how to identify these species when they're juveniles, because they don't look the same and they don't have mature reproductive organs. So we don't have as many ways to tell them apart. Males and females in some species are quite easy to differentiate because males tend to have a more pointed abdomen and females just tend to be rounded. And that does relate to their reproductive organs again, because males have a penis and it has muscle that attaches to the very sort of end of the abdomen. And so the shape of their body is basically to accommodate that. Females have quite a lot of eggs. So if they're gravid or it's late in the season, it's like August or September, females will be really big like this because they've got eggs ready to lay. And the way they lay their eggs is using what's called an ovipositor, and it almost looks like a worm that comes out of the front of their body, and the eggs come through that tube and are laid into the soil or into bark crevices. So this is the bigger picture of what I was showing before. This is a pair of a Japanese species, Libunum globosum, mating. So the male is over here and the female has this dark stripe. And so their mating is face to face, although they can't really see each other, of course, because their eyesight's not good. And their eyes are on the top of their bodies. So what's happening in here is that the male's using his pedipalps to clasp the female. This is actually the base of the penis. So intermission is happening, and the female's genital opening is also sort of oriented anteriorly at the front of the body. So if you see a pair of Opiliones and they're sort of face to face grabbing each other, that's what they're doing. And so mating can be as quick as sort of 30 seconds of sort of grab. Females, if they don't like a potential mate, will usually try to put their face down to avoid the male grabbing onto her. They're often much bigger, so they can kind of push a male away and run away. In this species, you can see the male has very large pedipalps. The female doesn't have those. So he's got quite a lot of muscle and leverage in order to sort of hold on to a female until she agrees to mate with him. So that's what's going on there, if you ever see that. These are species that you're less likely to see because all of these occur in Central or South America. But as I mentioned previously, there are a number of species of Opiliones that actually take care of their young, and both males and females are sometimes tasked with this. So in this species up here, this is the female. She's actually laying eggs, and so she's standing over them. So you can see there's a clutch, a couple rolled away. This is inside a little mud excavated turret of mud on the ground. And that's actually the male over here. So he's going to come in and sort of stand over them just like the female will do. He'll rotate these eggs and protect them from any predators that might come and try to take some eggs or lay their eggs, lay their larvae on top of the eggs. In this species, they don't actually occur in these colors. These are just paint markings to tell apart some different males. Both of these males are actually caring for eggs in these clutches down here. So these are on undersides of leaves in the Atlantic forest in Brazil. And these males will stand over them, they'll clean them. There's sort of a mucus coat on here that was secreted by the female when she laid them. But the female will come in, lay her eggs after mating with the male that's present. Sometimes she'll lay some additional eggs that were from a previous mating. And so I'm actually working with one of the researchers that studies these species and put these paint markings on here to do genetic work to figure out for every male that's caring for a clutch, how many of these eggs actually were fathered by him. So what's their sort of effort into their offspring? So that's a pretty unique behavior that we really just don't see in other arachnid species. And so that's really cool. So in the last couple of minutes here, I'm just going to talk really briefly about some research that I'm doing on Opiliones that's kind of off the beaten path of the research on reproduction and evolution of sex. And it actually focuses on biomaterials, so sticky proteins that different arthropods make. So, don't really try to read this because your eyes will cross and you won't get too much out of it. But what I'm showing up here is a figure of amino acid sequences. So these are subgroups that are repeated many times within the total sequence for proteins that are made by a type of spider that I'll show in a second. And what we know is that something about these amino acid sequences or their repetition or what is sort of modified in these proteins after they're transcribed in the spinnerets, if it's a spider, in the salivary glands of this velvet worm, for example. Some of those modifications might also be important to make proteins sticky. And so what we're doing is studying a bunch of different arthropods that make sticky proteins, and we're trying to understand what is sort of common about what they make. Is it the sequence? Is it the amount of repetition? Is it the certain amino acids that are found in the sequence? Or is it glycosylation, so sugars that are added to the protein after it's transcribed? So here's your chemistry for folks that are interested in that. The specific sugar that is added to sticky proteins in most cases that makes them sticky is called GalNAc, or N-acetyl-galactosamine. That's this yellow square here. So GalNAc is like added to certain amino acids, and it confers stickiness because it likes to sort of stretch with water. So it likes to be affiliated with water. And so a protein that might just sort of curl up becomes sort of swells up and becomes sticky. And the types of proteins that we think about mostly that have this activity when they're in the presence of water and they have these chains initiated with GalNAc are called mucins. So think about sort of the gross mucus in your nose when you have a cold. It's kind of sticky, you know, you want to blow it out because it's stuffing up your nose and you can't breathe very well. So that's actually just mucus that's kind of gone, you know, haywire. But mucus has really important functions. It's protective, and in a number of organisms that produce mucin-like proteins for prey capture, it's very important because it's what they use to catch their prey. So you know spiders produce webs that are sticky. So here's what a thread of the capture spiral of a spider looks like if we zoom in on it 30 times. And you'll see these little beads of glue on a core or a fiber of silk. So there's a non-sticky silk that these beads of glue are added to, and they can be added in different ways in spiders. So in Latrodectus, like widow spiders, they actually put their sticky glue droplets close to the bottom of the web, and they're trying to catch prey that are just walking around on the surface on the forest floor. In this species that Sarah, my former postdoc, who is now doing research at UNC Charlotte, we studied together. This is called a bolus spider. What it does is it actually doesn't really make a web. It just spins a little piece of silk and puts a whole bunch of glue at the end of it, and it whips that around and hits moths with it. So it hunts at night, and it's a moth specialist. Here's a velvet worm. They actually have glue cannons in their head, and they fire glue at their prey and incapacitate it with their glue. Here's an example of a dipteran, so a fly, a larval fly. These are called glowworms. And glowworms either will build sort of these chains of glue droplets, or they'll sort of make a little web for themselves and have glue droplets, and they use the glow in their bodies to attract prey that will get stuck to the glue that's on their webs. And lastly, the one I'm most excited about in the research that Sarah and I have just started to work on with these prey capture glues is, of course, an opilionid. And this is an opilionid that is called Sabicon. That's what the genus is called. There are a bunch of different species. They're not really the phylogeny is not well understood. But one thing all these species have in common is that their pedipalps have hairs on them, and each one of those hairs is tipped with a little bit of glue. So we call them setae. These setae are actually what, instead of a silk fiber like in the spiders, these setae are what the glue droplets sit on. And so it's almost like they've got two catcher's mitts, and then they catch their prey, which is usually collembolans or springtails. They grab their prey using their two sticky catcher's mitts. And then they use their chelicerae to cut up little pieces and feed them into their mouth. So that's kind of their feeding regime. And so we're really fascinated by what this stuff could be made of, what kind of protein it is. And so the way that we're planning to, so we recently got National Science Foundation funding to do this research on glue. Sarah and I did. And what we're planning to do is test using a force transducer, how much force we need to pull those tiny little droplets of glue off a surface. So how it functions, and we'll combine with that information about the sequence to learn more about glycosylation, protein sequence, and how those lead to making different kinds of proteins that are sticky. So I might have gone a little bit sort of over on time, and I appreciate your interest and your attention. I just want to sum up by saying a few things that we talked about throughout this presentation. One is that Opiliones is an order of arachnids that aren't spiders. They're not dangerous to people, although they can be smelly. They occur all over the world except for Antarctica. Some of them actually have caring fathers. Some actually make glue. And in many cultures, Opiliones aren't the scary sort of gross things that some people may have them out to be. They're actually lucky symbols of a good harvest, and they're great to have around. So, you know, if that was interesting to you, or if you find that you have additional questions, here's my email address over here. I tweet frequently about science-related topics. That's my handle at Dr. Sadie's. And this is my website where you can learn a little bit more about the research that we're doing and see some of the folks that are in my lab currently. And so with that, I'll just thank you all. And if there's time and interest, I'll take more questions. And I'll just show some clips of things from Japan.
Well,
all right, now you can.
That was great.
You can do it.
Bird. You can actually share.
Yeah, I will.
Yeah, can we come back if we do have some questions?
Sure. Oh, that's a relic slide from an old talk. Okay, all right.
Okay, and let me just put a spotlight on you. Thank you. The reviews are coming in and they're all fabulous. Wonderful, wonderful, wonderful. We do have some questions, and one is that we're going to get to. It was, how come sometimes you see them in large numbers hanging out together like in a shed, and sometimes you only see one at a time?
Yeah, it's a good question, and it's something I don't think any of us know for sure. And the answer, or one of the many hypotheses, is that because these daddy long legs have a lot of surface area, so they've got long legs, they lose a lot of water through their cuticle. You know, they don't have lungs, so their breathing, it takes place through sort of tiny holes in their exoskeleton. And so when they breathe, it's passive, and they're losing water just by having those sort of openings in their body. It's almost like if you walked around with your mouth open all the time, you would have a dry mouth, most likely. So one of the hypotheses for why we tend to see those large aggregations is that they're all sort of hitting upon the same habitat, and they're conserving moisture by kind of raising the microclimate humidity. That needs to be tested. Another reason, so what we notice about these aggregations is that rarely is it just like a whole bunch of individuals mating, or a whole bunch of individuals feeding on each other. Mostly they're just sitting around, and they only kind of get startled and run away if somebody goes in there and pokes them or whatever. And so another reason is that they do, when they are when they're shedding or when they're molting into their adult stage, they do tend to find the same sort of environments to molt on. So they tend to group up in corners of bathrooms out in the woods, or, you know, up in these sort of crawl spaces, because it's a convenient area that they all kind of hit on at once, and they're all, you know, reaching that molting stage around the same time, and they want to get their molt ready and get to mating, right? So but it does seem to be relevant that this humidity hypothesis, because their molting, requires them to be a little bit moist. If they're too dry, they'll usually die. Their molt will not be successful. Folks that, have kept even, reptiles, for example, or spiders as pets, they need a more humid environment, especially if they're about to shed, because if it's too dry, it's irritating to them. And actually for arachnids, they can die if they have an unsuccessful molt, because a lot of people don't know this, but I get excited to talk about this, sorry, but the pharynx, or the mouth and throat of an opilionid is actually coated in cuticle, so it's hard, and that's sort of a safety measure, right? Because they have to cut their food into little pieces. They don't want to kind of scrape going down. However, their brain is sort of kidney bean shaped, and it's situated around the pharynx, which means when they go to shed, they have to shed all that pharynx. So they basically have to throw their mouth up through their brain to get that covering off the anterior part of their body. So that's dangerous. If you can picture that, it would be, if you don't get good detachment or whatever, you just die. You can't get out of your skin and you just die. So that seems to be probably relevant to why we see these large aggregations. It's a great question though, and something that no one really knows for sure.
Virginia asked, where do they lay eggs? You did show some pictures of a couple of spots, but in Maryland,
mostly in mosses. So they like wet and places that aren't going to get hit with too much fungal growth. So and that's where the columbolins can actually be kind of helpful, those environments, because they like to eat fungus. So they like sort of shallow, moist-ish soil, or bark crevices deep in there, because it's got to stay dry and it's got to stay protected, sometimes over a winter. So is it easy to find these eggs? No, not in the wild. When we keep them in the lab, we usually give females a dish of dirt that we spritz with water to encourage them to lay their eggs on that.
And I did want to mention to everybody that Dr. Stellwagen, did I pronounce that correctly?
Yeah, you did. Yeah, she presented to the Society before about her research on silk. So she's the one, she worked in my lab for 3-ish years, and she was the one that was doing and continues to do work on silks. So we got a collaborative grant funded this year that we're really excited about, and that's going to continue to allow us to do work on sticky proteins in different.
Right. So for anybody who wants to learn more about that, her presentation is on our YouTube channel, so you can go back and take a look at what Sarah had to say about that, her research. And speaking of that, Jillian says that ortholasma also use glue for capturing columbella.
Yeah. So there's actually, Jillian, I recognize your name. Thanks for coming to this talk. There are a number of species that have these glandular setae. So all the Hespero, Nemastaba, is that the group? So Tyracus, Sabicon, Ortholasma, they have these sort of cetaceous palps with the glue. So we've decided to focus on Sabicon because we have species on the East Coast. We have one Sabicon on the East Coast and several species on the West Coast that we can collect and compare. And they're a little bit bigger and a little bit easier to find. I went to Portland, Oregon to collect Sabicon in October, and they're different than the daddy-longlegs species that I'm used to. They're not easy to find. You have to kind of dig around, and I'm still not really clear on why they choose certain sites or why they tend to be found in different sites, but they're awesome looking. I've got a couple students that have collected glue off the CD that we're going to be able to use to do some work.
Tom asks, I mean, since they are not venomous, how did that myth come to be? Do you know?
You know, I have a hypothesis for it. I'm not sure if this is correct, but I know that in scorpions, there are some quite small species that do produce medically significant venom. So that basically just means like it could make you sick, or it could give you a severe injury were you to be stung by one. And some of those scorpions, as I said, are kind of small. And so I wonder if maybe that sort of common logic developed over time to be, you know, things that are small can be perhaps even more venomous or perhaps even more dangerous to us. I'm not sure why that came about, but you know, it is the case that unless you have sort of a hand lens or really good eyes, right, it's not always easy to see all those little organs, all those little mouth parts and palps. But you know, you do kind of get used to handling them and taking a look at them. And so I encourage everybody, you know, in the spring when you start to see some of these out, just pick one up. You know, just pick one up and take a look at it. You know, you're not going to hurt it, and you know, you'll perhaps impress a loved one in the process. So.
Do they prey? Sam wants to know, do they prey on each other? And I think you said that they did.
They do. Yeah, they do. They can. You know, particularly if they come across an individual that's, you know, wounded, dying, they'll probably, you know, be sort of open to that, just because they'll eat what they can get, right? You know, they live fast, hard lives. So so they would eat each other for sure.
And Richard wants to learn more about your research on sexual reproduction. Where could he do that?
I, on my website, have little blurbs about some of the ongoing research projects. And I also have links to all of my papers on the publications tab of my website. And I will note that not only do I have sort of the sciencey, you know, scientific peer-reviewed research papers, I've also written a couple sort of, you know, broad interests papers. One focuses specifically on asexual reproduction, which is something we study in the Japanese species. So if you're interested in that, the link to that is on my website. It's in the conversation. So that's free. You can take a look. Hopefully you're interested. Yeah. And
Gail wants to know, how did you first get interested in Opiliones?
Yeah, you know, I didn't seek to. There's a lot of people who go into graduate school in ecology and evolution that want to study specifically a certain organism, like wolves, you know, or birds. I was more interested in invertebrates than vertebrates. I kind of always have been. And I thought, you know, I grew up in a mixed race family where, you know, it was very important to sort of, you know, get your education and kind of give back to the community, show your skill. So I thought I would probably go to med school when I went to undergrad. And then I took a class in comparative physiology. So we talked about, you know, how do humans circulate blood in their bodies, but also how do wolves do that? And how do, you know, I don't know, mollusks do that? How do squid and worms do that? And somewhere along the line in that class, I just realized I couldn't go to medical school because I was just too interested in the diversity of life, especially in the diversity of body plans and life forms within invertebrate groups, just because there's so much more than invertebrates and there's so many understudied things. So just being able to study and compare how like life's problems like eating, reproducing, and survival are accomplished in different orders, to me was much more fascinating than just focusing on people. So when I applied for graduate schools, I applied to labs that were doing research on reproductive behavior and evolution in invertebrates, and the lab that the person that I clicked with most became my PhD advisor. And so that kind of stuck, for better or for worse. But broadly, I really enjoy all types of arthropods, not just Opiliones, but there's a lot of opportunity to just teach people new things because it's not a group that gets a lot of interest. But there's so much opportunity because they're so common here. So it's worked out nicely, I would say.
Well, this has been wonderful. Thank you. And I hope that you will consider coming back and sharing some more knowledge with the society and our community of the curious, because we're always anxious to learn from such people.
Yeah, it's my pleasure. And I look forward to when there's an open day, check out the collection there, because I love taxidermy. I enjoy producing it. And I would love to check out what's there. So thank you for inviting me.
Well, you have an open invitation and we'll take you down to the basement to see all the collections. It would be wonderful.
Great. All right.
Everyone, thanks for joining us. And we hope that we'll see you in next week for the spotted lanternfly with Dr. Mike Ropp, and then ending up with rattlesnakes. So we're giving you all kinds of information to share, and not controversial information to share at the family holiday table. You can impress everybody and have a great time. So come back and learn with us, and everyone stay safe and stay curious.
Thank you so much. And thanks for